Hey everybody, welcome back. I hope that you guys enjoyed your break. Hope that you got some time to relax and everything like that. Um, it is the 17th and I am ready to go as evidenced by posting the PowerPoint 4.5 minutes ago. Um, so as I shared with you guys, my wife and I welcomed a baby boy and I wanted to share a picture. Um, and I This is Hank is his name. He's delightful. He's wonderful. We are very, very thankful to have him. He's wonderful. Thank you very much. We're very excited. It's really a wonderful thing. Um, so there were a couple of let's see. Um, this week in our class, we're going to talk about kind of bringing things back together with respect to sugar metabolism. Um, and so we're going to talk about today gluconeogenesis. Um, I don't think that this is going to be, how should I put it? Um, I don't think it's going to be too cumbersome. I don't think it's going to be something where you're like, oh, gluconeogenesis. I'm hoping that you'll walk away from it and you'll be like, okay, cool. I feel pretty good about that topic. Um, Now, with that said, I was looking at the semester schedule, and I think we have it all told. I think it's like from today until May 5th, that's 8 weeks. Um, so the semester is kind of like it kind of closes in pretty quickly. Um, and with that said, uh, anything that I can do to help you out, I'm going to work hard to do that, to support you, get you the information that you need sooner rather than later. I apologize for any delays in grading and stuff. I want to catch up on that. I think that your activities are graded up to date, but I will do a better job. With that in mind, I wanted to go ahead and jump into gluconeogenesis. And the reason why the way that I like to do this is I like to talk about glycolysis as kind of your forward pathway. And then I like to talk about the pentose phosphate pathway as kind of an offshoot. And then I like to talk about gluconeogenesis because it's basically the reverse of glycolysis. Um, so in essence, I've got this slide right here, or I've got a nice little summary slide that captures sugar metabolism as a whole. In this class, we're not going to tackle all of this. In this class, what you should expect to kind of walk away with is I want you to walk away with kind of I would say like a working knowledge of this right here, a pretty good idea of this right here. Then what we're going to catch up on today is going to go here. And then before the end of the semester, we're going to send things down this way. So I'm really happy that I made a somewhat confusing pathway even more complicated with drawing over it. So um, just when you didn't think it was possible, I did it. I made something less clear. I apologize for that. Um, so if you're thinking about the pathways that we've looked at with respect to glycolysis, pentose phosphate, and what we're about to tackle, which is gluconeogenesis, um, in terms of what should you prioritize as the one that you're going to kind of get the strongest understanding of, I would recommend glycolysis. Gluconeogenesis and pentose phosphate are a little bit more big picture. Um, but I think that it's also helpful to remember and look at gluconeogenesis as kind of like a nice support for glycolysis because it's doing the exact opposite. So essentially, that's what we're tackling. Glycolysis and gluconeogenesis. Gluconeogenesis is undoing the work that was done through glycolysis. Now, a fair question for that is, well, why? Well, one of the important parts about this is, and this is a challenge with any sort of biochemistry class. Um, if you've ever heard the terms biological chemistry, you might think like, oh, okay, well, that's the chemistry of biological molecules. You're right. And that is the study, the chemical study of biological molecules. Biochemistry and biological chemistry, there's like very fine, subtle differences. Now, I think that it's very common for people as biochemists to be like, oh, well, I'm interested in understanding how this organism works. And then they just kind of they try to almost like divorce themselves from the human connection of biochemistry. I am not that person because I think that it's kind of like understanding sugar metabolism is fundamental to a human's existence. Um, and so, with that in mind, glucose is a primary energy source for humans. Regulating blood glucose levels is a fundamentally important part about uh, a human being's life and livelihood. Um, so, just as we use glucose and we break it down through glycolysis, we also sometimes need to build it back up so that we have a certain concentration of glucose in our bloodstream. Now, in some cases, what we're doing is we're just taking pyruvate, the product of glycolysis, and we're using that to go back towards synthesizing glucose. Now, the formation of new glucose molecules can be done from non-carbohydrate precursors. For example, lactate, pyruvate, and glycerol. Now, that note right there, non-carbohydrate precursors. I think a reasonable approach to that is to ask, well, what does that mean? That implies these three molecules are non-carbohydrates. Okay, got it. What do you mean by formation of new glucose from, well, a carbohydrate precursor? Well, something like glycogen. Glycogen is that molecule that's made up of thousands upon thousands of glucose molecules. Well, you can take that and break that down into individual glucose molecules. Glycogen is a carbohydrate. So when you see that non-carbohydrate precursor, I think it's also helpful to think about, well, what's the precursor that is a carbohydrate? Um, so in addition to these big three, there are also some amino acids that are used for making sugar. Does anyone know? And I don't know that I think I maybe said the word one time in transition and like very temporarily. Um, what are amino acids that you can use to make glucose? If you maybe you've heard that, I'm not too sure. If you've heard it, go ahead and add it to the chat. Take a chance. If you want to add it to direct message, welcome to. What I'm looking for there is glucogenic. There are some amino acids that are glucogenic, so they can be used to make glucose. Um, that is not something I have expected you to know or anything like that. I think I maybe mentioned that. If I didn't, well, then I apologize. But now I have. So there are some amino acids that you can use to synthesize glucose. Now, gluconeogenesis, again, this is that human component of it. Where does it take place? Well, it takes place primarily in the liver. And, well, under certain starvation conditions, the kidney can also contribute and make a small amount of glucose. Now, gluconeogenesis. Between meals, blood glucose levels are maintained by the hydrolysis of liver glycogen. When liver glycogen stores are depleted, during prolonged fasting or a vigorous exercise. And whenever like liver glycogen, we're talking about an intense or a vigorous exercise like uh, an endurance race of sorts. The gluconeogenesis pathway provides your body with glucose. So it's kind of that safety net of sorts to make glucose so that the sugar level or the glucose level in your bloodstream is kind of maintained. Now, this is very important because your brain and your blood, or your red blood cells, are highly dependent on glucose. Okay, so you've got to support that. You've got to maintain this kind of consistent and steady flow of glucose, well, for these pretty darn essential organs. Now, gluconeogenesis as a pathway, this is one thing that I don't know why, but as a student in a metabolism class, I really failed to understand this, and I take great shame in this. If this is something you don't know or are still working on, you do not need to take shame in this or anything of that nature. But I remember just being like, man, gluconeogenesis, how on earth does that relate to anything? Got glycolysis, good, but that's it. Gluconeogenesis, what? Well, when your blood sugar levels are low and liver glycogen is depleted, 7 of the 10 reactions of glycolysis are reversible. There are those 3 that are irreversible. We've got hexokinase, phosphofructokinase, and pyruvate kinase. Those are our 3 irreversible reactions. That means that they'll go one direction, but not the other. And so the 3 steps that are not reversed are their 3 irreversible steps. They are hexokinase, PFK1 or phosphofructokinase, and pyruvate kinase. Now, there was a term or a phrase that I used for PFK1. Yes, it's irreversible, but what is a unique detail about PFK1 that's important for you to know that makes its irreversibility more kind of special, I guess? Add it to the chat. First committed step. Thank you. Great job. It's not the first step, it's not the first irreversible step, but PFK1 is your first committed step. That means we're going through glycolysis. Great job, everyone. That's an important distinction, an important thing to kind of separate. Now, our 3 irreversible reactions of glycolysis are bypassed in gluconeogenesis. So in gluconeogenesis, those 3 enzymes, hexokinase, PFK1, and pyruvate kinase, don't do anything. Because what we need to do, do the opposite of it. Absolutely. It does. Perfect. Well done, Brianna. Now, here is another display of our pathway through glycolysis. This is our forward. Glucose on down to, which glucose is 6 carbons, down to a 3-carbon pyruvate and another 3-carbon pyruvate. Okay, so what we need to do is offset those 3 reactions. Everything else, it's fine. Glyceraldehyde 3-phosphate can interconvert between itself and DHAP, or dihydroxyacetone phosphate. So those 2 can go back and forth. Now, the rates of these vary a little bit, but they're still going to be able to go back and forth. So everything on here is reversible. Now, except for the highlighted 3 reactions. So we need to overcome those. Now, we've got to have 2 molecules of pyruvate. This is one of the places where I like to think about, okay, I like to do the kind of rough accounting of carbons. We've got 2 3-carbon pyruvate molecules. Those 2 molecules have to be used to get to our total of 6 carbons that make up glucose. Now, in order to produce glucose from those pyruvate molecules, we've got to bring in 2 NADH, 4 ATP, 2 GTP. If you think back to what was made in the process of going from glucose to our 2 pyruvate molecules, well, we made 2 NADH. We made 4 ATP, but it was a net ATP because we also had to invest. So then we also actually have to bring in this 2 GTP molecules. So just a second. It's just a second. Sorry, everyone. Sorry about that. That was my 4-year-old son, Arlo. He is sick. He's got, I think it's possible that he's got RSV, which sucks. He's like, yeah. Um, but he could not find the remote. And then he found it exactly where he put it. So he got me. I apologize, everyone. And he also apologizes. Okay. So what we've got is our components that we actually have to, fair enough, that we have to invest. Now, when it comes to gluconeogenesis, what I really want you to do and what I think is really helpful is to think about what matters in glycolysis. Our 3 irreversible steps. Yeah, there's lots of other details there, but those 3 irreversible steps are what we have to do to offset glycolysis, and that's what we need to do to go through gluconeogenesis. So I think that it's helpful to kind of look at each of these individual reactions and say, okay, I need to get from here to here. How am I going to do that? Okay, so we've got our Y. Our Y is to kind of help maintain a certain blood sugar level. Now, the first molecule that we're going to use, our first non-carbohydrate uh, metabolite, is pyruvate. We get our pyruvate, and I'm going to use this model right here because this is a nice figure of a cell. And this is my kind of figure of a cell because there's a handful of wavy lines and they're like, yeah, that's a structural protein. That's a porin. How much details there? Enough. Enough details there. That's what we got. Okay. So I think that this is also helpful because it gives you a sense of where's this taking place? Where are these different things taking place? And when you go on to take a metabolism class, that becomes very important because you have these kind of competing pathways that, well, one is going in this direction, the other is going in this direction. Well, how are they not just constantly resetting one another? And when we have a example of a eukaryotic cell, like what we've got right here, we can identify where different processes are taking place. So I'm going to go ahead and bring your attention to this area right here, this kind of, I would say, light khaki colored region of the cell. That's your cytosol. And in your cytosol, that's where glycolysis is taking place. And I'm going to highlight this glucose right here, goes all the way to our pyruvate molecules. That is glycolysis that's taking place in the cytosol. Now, what we need to do in order to get to gluconeogenesis is actually, well, we need to, if we're going from pyruvate upstream, what we need to do is we need to get pyruvate converted to phosphoenol pyruvate. Because our last step, I'm going to call it step 10 of glycolysis, takes pyruvate and converts it to, or sorry, PEP, phosphoenol pyruvate, and converts it to pyruvate. So that's our last step of glycolysis. So effectively, our first step of gluconeogenesis is to get from, well, pyruvate back to phosphoenol pyruvate. So in glycolysis, that's a single enzyme that does that. It's a single enzyme that removes that phosphate group from phosphoenol pyruvate to make pyruvate. or PEP to pyruvate. Well, in gluconeogenesis, it's not that simple. It's not that direct. Instead, what's happening in gluconeogenesis is we first have to take pyruvate and put it into the mitochondria. Now, the mitochondria is indicated by this whole thing over here. This is our powerhouse of the cell. And in this kind of magenta colored or yeah, magenta-ish colored region of our cell, that is basically where the most important action of the mitochondria is going to take place, where most important processes are taking place. That's within our inner mitochondrial matrix. Now, what we need to do in order to get the wheels turning for gluconeogenesis is we have to get pyruvate from the cytosol into the mitochondria. Now, how is that taking place? Well, it's taking place with the help of this right here, MPC. And MPC is a mitochondrial pyruvate carrier. Think about this as a protein that is able to kind of survey the area and look at a molecule, and it's able to pick something up and be like, oh, okay, well, you're glucose, not interested. Oh, you're fructose, not interested. Oh, you're pyruvate, great. I'm going to move you from this side of the membrane to the other side of the membrane. It's almost like a bouncer for a club. Now, in addition to that MPC, you probably noticed this right here. That that is that's our outer mitochondrial membrane, and that membrane is a little bit more porous. This is not an important detail for you right now, but again, as you go into metabolism, this is going to be a very important part. Pyruvate getting across this membrane can do so fairly easily because that membrane is not super selective, but there are also proteins known as porins that are semi-selective, and they can just kind of accommodate specific molecules or molecules of a certain range of size as they move across the membrane. So if you're thinking, okay, what's the first step if I want to initiate gluconeogenesis? What I want you to walk away with is I want you to think, okay, I've got to get pyruvate into the mitochondria. That's my first step. Because it is in the, or if it's in the cytosol, that's the wrong place for gluconeogenesis to take place. I've got to get it into the mitochondria. Okay, so this right here is our first step. Pyruvate moves into the mitochondria, and lo and behold, it's still pyruvate. Now, once that 3-carbon pyruvate molecule is in the mitochondria, we're right here. What we need to do is, and if you look ahead, we need to go from here, pyruvate. What was the molecule that we were in glycolysis? What becomes pyruvate? PEP. If you look through all of these different molecules, next is oxaloacetate, malate, malate, oxaloacetate, PEP. Whew. We've got a couple of different steps that are required to go from pyruvate to PEP. So when you see that right there, pyruvate going to phosphoenol pyruvate, there's a word that comes to my mind that I think is really important here, and that is regulation. Going from pyruvate to phosphoenol pyruvate, there's a couple of different barriers. There's a couple of steps, deliberate steps to get us from pyruvate to PEP. So that's not just a rubber stamp, okay, moving forward. So pyruvate goes to PEP, but it takes a little ways to get there. Now, the first of the steps that we're going to look at is we're going to look at pyruvate being converted to this molecule known as oxaloacetate, also known as OAA. Okay, so step number 1, we're in the mitochondria. Pyruvate goes to oxaloacetate. Well, the enzyme that does this, is known as pyruvate carboxylase. We've got 1, 2, 3 carbons. And what we're going to do is we're going to use ATP and we're going to make ADP and PI. Okay. ATP is a high energy compound. If you look at oxaloacetate, add to the chat is that add a Y to the chat if it is a phosphorylated molecule and an N to the chat if it is not. Oxaloacetate. You're right. It's not phosphorylated. Okay, so essentially what we did is we're using ATP, but we are using kind of the fuel or the energy harnessed by ATP hydrolysis. And what are we doing? We're taking this right here, HCO3, to add CO2 onto pyruvate to make oxaloacetate. So this enzyme, pyruvate carboxylase, we carboxylate pyruvate. So we add CO2 onto pyruvate to make a 4-carbon long molecule known as oxaloacetate. Now, this step is metabolically irreversible and it is taking place in the mitochondria. Now, that oxaloacetate, once we've got that, that's an important step because now what we can do is convert it to malate, and malate is a molecule also known as malic acid that is going to go through a couple of steps, go through steps known as a malate shuttle. So this is a molecule that can be kind of moved from one side of the membrane to another. So what we're going to do is effectively we're using malate as our intermediate to get oxaloacetate out of the mitochondria. So what we did in a nutshell was one, moved, I'm going to say PYR for pyruvate into mito. Okay. Next, carboxylated. Carboxylated PYR to make, or I'll say into, and then the street name for oxaloacetate is OAA. I like to use the phrase street name because it sounds like I'm a narcotics agent in a movie or something like that. So, if you ever meet a narcotics agent, see if they respond to OAA. I don't know that they would. Um, anyway, we carboxylated the pyruvate to make oxaloacetate. Then I'm going to, like I said, for this class, we're not going to take a deep dive into this because what we're going to say about this is we're going to move OAA out of mito. Mito. Yes, there is a shuttle involved in the process, but what I want you to know is I want you to know that the carbons that make up oxaloacetate get out of the mitochondria. That's the important part for what I want you guys to know. The movement, the actual steps that are currently highlighted, this malate shuttle, when you take metabolism, you'll take a deeper dive into it. But for our purposes, I want you to understand that these three steps are the important parts about getting oxaloacetate so that we can use it outside of the mitochondria. Now, in some species, in some organisms, PEPCK, PEPSIC, is found only in the cytoplasm. Oxaloacetate must be transported out of the mitochondria. We did that. Now, you're probably looking at this and saying, oh, great. He forgot a slide because he introduced an acronym and said it, but didn't say anything about it. PEP is phosphoenol pyruvate carboxy kinase. So phosphoenol pyruvate carboxy kinase, or PepCK. Again, that's kind of like OAA. Talk to some narcotics agents, and maybe they'll be like, "Oh yeah, Pepsi, got it, Pepsi K, I know." Um, that is our cytosolic protein, and what it's going to do is it's going to deal with that oxaloacetate and convert it to our phosphoenol pyruvate. Now, this is a little bit of detail into how we're doing that. But again, what I want you to know is that oxaloacetate, we get that out of the mitochondria. Okay, you can answer later. This is a very late question. I can set the Okay, so I got a question from someone and they told me I could answer it later, but if I say that I'm going to answer it later, I will inevitably forget, and the later will be in about 3 to 4 weeks, so I'm going to answer it right now. Um, I can't stop thinking about it. Earlier, we used GTP. Is that guanosine triphosphate? Yes, it is. And if so, why would we use GTP over ATP and vice versa if they're both high energy purines? They are both high energy compounds, and that's a great observation. The difference between them is that an enzyme is going to have an affinity for ATP and GTP. Different enzymes have different affinities for ATP and GTP. Chances are that it evolved to use GTP versus ATP. And if it was only provided ATP, it might use ATP. One of the reasons that I always think about why we have those, or why those different nucleotide triphosphates exist is, well, it is a regulatory step almost. Um, if the GTP concentration is really low, that enzyme's not going to be as active. So I always kind of think about it as like a built-in regulatory mechanism. I hope that answers the question. So if it does not, please follow up. Um, okay, so like I said, oxaloacetate converted to malate by the mitochondrial malate dehydrogenase. And then malate is transported across the mitochondrial membrane. Malate is converted back to oxaloacetate by a cytosolic or cytoplasmic malate dehydrogenase. Um, and then oxaloacetate is then converted to PEP by phosphoenol pyruvate carboxykinase. The important thing that I want you guys to know, and I'm going to go back and add my fourth item here. OAA converted to PEP by PEPCK. So I'm gonna come back to this slide periodically so I can keep kind of a running log of important items for you guys. Okay. Dun, 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 dun. Okay, and this is where I want us to be. This is effectively our important step of getting back to one of our glycolysis intermediates, because oxaloacetate, was that in glycolysis? No, it's not. Phosphoenol pyruvate, however, is within glycolysis. So when we go from oxaloacetate to phosphoenol pyruvate, catalyzed by phosphoenol pyruvate carboxykinase, we're driving this with the help of GTP, guanosine triphosphate. This is metabolically irreversible. So if you're thinking about this in the context of glycolysis, what are we working on? We're still undoing reaction 10. Reaction 10, also known as pyruvate kinase. We're still offsetting that. Well, let's look at this. What we do is we take our oxaloacetate, or OAA. We use GTP, guanosine triphosphate, and we are going to decarboxylate it. We're going to remove that CO2 that was added in the previous step to make oxaloacetate. And what else are we going to do? We're going to pluck that CO2 off, and we're going to throw a phosphate on to make phosphoenol pyruvate. Now, is this like, I think about this as another layer of regulation. This is another layer that helps make sure that glycolysis is taking place and it's not always being undone by the action of gluconeogenic enzymes. Because we have two enzymes effectively, or two major steps that offset this. We have to convert pyruvate to OAA, then OAA to PEP. We're not just flipping the coin back over. Instead, we have to go through this kind of deliberate process. Okay. Now, once you get through there, we are effectively 33% of the way done offsetting glycolysis because of our three enzymes, pyruvate kinase being our last one. Now all that we need to do is actually what we've just been talking about is really going and taking care of this step at the bottom. Once we get phosphoenol pyruvate, we're golden. We can go all the way back up to fructose 1,6-bisphosphate. So all of these steps are reversible, I'm sorry. Once we get to fructose 1,6-bisphosphate, sorry for that noise. I wish I had the like I wish I had a mute that I knew how to use properly. Um, okay, so what we need to do is we need to offset fructose 1,6-bisphosphate, or the production of fructose 1,6-bisphosphate. This is our PFK1 enzyme. So PFK1, when we're talking about glycolysis. Now we're going in the opposite direction. And this is kind of a good a good general rule is that a kinase is a type of enzyme that's going to add a phosphate and use ATP as a phosphate donor. That's pretty common. That's not always the case, as is evidenced by pyruvate kinase. But most often, when you see kinase, think about, okay, we're adding a phosphate. Now, the opposite of a kinase typically is this, a phosphatase. So fructose 1,6-bisphosphate is converted fructose 6-phosphate by fructose 1,6-bisphosphatase. Now, this step, where we are going from this molecule right here, our doubly phosphorylated fructose molecule, to a singly phosphorylated fructose molecule, fructose 6-phosphate. So fructose 1,6-bisphosphate to fructose 6-phosphate. Well, the type of enzyme that does that is a phosphatase. And this is an example of, well, biologists and biochemists being biologists and biochemists as opposed to literature majors. They said creativity, ha! This is a phosphatase. What does this phosphatase act on? It acts on fructose 1,6-bisphosphate. So we'll say fructose 1,6-bisphosphatase clips the phosphate group off of carbon number 1 and makes inorganic phosphate. This hydrolyzes it. So it's not just making ATP. Instead, it's removing an inorganic phosphate as a part of this. Now, does anyone remember, and you can add to the chat if you remember this, what is the enzyme that precedes PFK1? Add it to the chat if you know it. We're going from fructose 6-phosphate to, well, I'm thinking about this. Sorry, let me rephrase my question. When we're thinking about glycolysis, I like the thinking face emoji. The first enzyme, hexokinase. Second enzyme, what is this one right here? It's our isomerase. Or yeah, so we're isomerizing that glucose to fructose. So we could say gluc to fruc. Okay, so that's a reversible enzyme. That isomerization, no big deal. Now we're at a point where we have to undo the action of hexokinase. Using, or yeah, the enzyme that catalyzes that reaction, lo and behold, a phosphatase. That phosphatase is acting on glucose 6 phosphate. So it's glucose 6 phosphatase. Also hydrolyzes this phosphate group off, releasing inorganic phosphate and glucose. This enzyme is unique because it's only found in the liver and the kidney, and it's kind of getting us back to the point where we have glucose. So these are all of the important steps of gluconeogenesis. So when you think about gluconeogenesis, there's basically 3 steps, but one of those steps has 2 parts. This is what's required to go from pyruvate all the way back to glucose. We got to go into the mitochondria to go out of the mitochondria. Then we've got to go make our 4-carbon molecule into a 3-carbon molecule, and then dephosphorylate, remove phosphates from a couple of different molecules. Now, for other types of molecules, so several metabolites can serve as precursors by the liver to form glucose. So the liver works very hard to make sure that you have adequate glucose in your bloodstream. Now, there are a couple of molecules that are also involved in the mix here. They are lactate, glycerol, and alanine. So we just went through pyruvate, getting pyruvate all the way back to glucose. What do these other molecules do? Well, lactate. This is a gluconeogenesis substrate. We'll refer to it as gluconeogenesis substrate level or number 1. Lactate can be released by red blood cells and other cells lacking mitochondria or with low oxygen concentrations. In the Cori cycle, lactate is released by skeletal muscle, and once the lactate reaches the liver, so when your lactate actually makes it all the way back to the liver, it's converted to pyruvate by lactate dehydrogenase, and then to glucose by gluconeogenesis. So what I want you to take away from this is that lactate, by way of the Cori cycle, can go back to make glucose. Now, the process is basically the same, but with one added step. Lactate goes directly to pyruvate. So what I want you guys to know about this is lactate uses the Cori cycle to make pyruvate. Okay. Now, next up, our second gluconeogenesis substrate. Glycerol obtained from degradation of fats in adipose tissue. Okay, so glycerol can be used to make glucose, but it's a little bit of a different way. So this is transferred to the liver through the blood. Once it's in the liver, glycerol is converted to dihydroxyacetone phosphate, which enters gluconeogenesis. Okay, now this is a sort of question that I really like in I think a good exercise. If we have steps 1, glycolytic steps, or I'll say glycolysis steps 1, 3, and 10. Step number 1 being hexokinase. 3 being PFK1, and then 10 being pyruvate kinase. If you plug glycerol in and you use glycerol to make glucose, do you use all of those steps? Do you use 1, 3, and 10? And a little bit of a spoiler, the answer is no. Now, my follow-up question to that is which enzymes are bypassed, or which of those steps are bypassed if you're using glycerol to go to glucose? And so, which steps, 1, 3, and 10, or 1, 3, or 10, are any of those steps skipped over if we're going from glycerol to glucose? And think about this right here. This dihydroxyacetone phosphate is what we're plugging into glycolysis. Where do we remember dihydroxyacetone phosphate being produced along the path of glycolysis or through glycolysis? Where is dihydroxyacetone phosphate generated? That is, it is DHAP. RP, you're absolutely right. So when you think about dihydroxyacetone phosphate being made, this is one of our two products. So I'm going to say one of two from aldolase. The other product of aldolase is glyceraldehyde 3 phosphate. So that's kind of in the middle of glycolysis. So if you use glycerol and that makes dihydroxyacetone phosphate, you're never going to make phosphoenol pyruvate. You're kind of plugging this dihydroxyacetone phosphate in the middle of gluconeogenesis and saying, okay, go back. And I'm going to go back to that slide where I had it all. Okay. And now I'm going to erase these writings. So okay, so here is glycolysis. And I'm going to say glycerol, do it over here. If you use glycerol, what that's going to do is that's going to be converted to dihydroxyacetone phosphate. That dihydroxyacetone phosphate will be able to make a glyceraldehyde 3 phosphate molecule. And so let's imagine that you've got two glycerol molecules. One's going to make this, one is going to make this. Aldolase is going to take those two to make fructose 1,6-bisphosphate. Then what's going to act on that? If we're going back to glucose, well, our phosphatase is going to act on it. Then we're going to make fructose 6-phosphate. Then we're going to make glucose 6-phosphate. And then our phosphatase is going to act on it. Okay. Answer honestly, did any of that make any sense? Okay. So what we've been going through over the course of today is getting pyruvate, understanding how pyruvate can be used to make glucose. Does that make sense? I will. I will get there. But first and foremost, pyruvate going to glucose. Does that make sense? We need to do that. And the why. Okay. So now, if you have a high concentration of pyruvate in the cytosol. Your body's going to funnel some of that pyruvate in the cytosol into the mitochondria so you can go and produce glucose. So that's like this is our base understanding of gluconeogenesis. Now, the next thing is, will you always have that pyruvate available? Not necessarily. Sometimes, rather than having pyruvate, you'll have something like lactate. For instance, if you're doing sprints, what's going to accumulate in your muscles? Lactic acid, also known as lactate. What you want to do is you want that to go away because that doesn't feel very good. But what eventually is going to happen with that lactate is lactate can be converted to pyruvate. And as soon as it's converted to pyruvate, and you have more than one pyruvate molecule, that can be converted through all those steps that we described to glucose. Does that make sense? Okay. So now what we're looking at is we've got two different molecules. Pyruvate, that's kind of our mainstream, our main line conversion from two 3-carbon molecules to glucose. Our next molecule is lactate. Well, lactate largely goes through the same process as pyruvate, but it's just got a couple of extra steps because we got to go from lactate to pyruvate. Okay, now I'll leave that. I'll leave lactate right here. Okay, so this is you're doing sprints, you've accumulated lactic acid in your muscles. What does your body do to compensate and to offset that? Is well, it's going to eventually convert some of that lactic acid or that lactate to pyruvate so you can regenerate some glucose. Now, the next thing is, let's say that you have decided you've given it a lot of thought and maybe you talk to a dietitian and they said, you know what you need to do is um, you need to modify your diet so that you have a high concentration of fats, a high fat diet, reduce your number of carbohydrates, increase your fat concentration. And you're like, okay, well, that sounds good. You're a dietitian, you know what you're doing. Now, fats are molecules that come in a couple of different forms. One form is as a fatty acid, but another form is as a TAG or a triacylglycerol, or also known as a triglyceride. Now, if some of these terms are like foreign, well, great news. We're going to get into them in this class fairly soon. But in this term right here, triacylglycerol, that is a molecule that looks like this. Um, Actually, I'll just draw, there we go. Glycerol. There we go. That really doesn't look like I spelled glycerol at all, but that's what I intended to spell there. So it's a glycerol, and it's got this Fa or a fatty acid chain. Here's another one, Fa, Fa. Okay, now that is maybe an unfortunate placement for a drawing over other words, but that's a triglycerol, a triacylglycerol. That's what some fats are going to come in. So if you eat a steak and it has lots of fat on it, chances are you're going to ingest some of those. Now, we're taking this dietary thought about this. Let's say hypothetically that you haven't eaten any sugar at all. What's going to happen to your blood glucose level? Well, it's going to dip. But what's your body going to do? It's going to find a way to get you glucose so that your blood glucose doesn't dip. Does that make sense? Okay. Yeah, absolutely. Now, what your body is going to do with this molecule, a triacylglycerol, is it's going to chop these fatty acids off. And I'm going to say this, FA, another FA, FA. Those are all released. What is left behind after those fatty acids are chopped off? Add it to the chat. Glycerol. So now glycerol is a 3-carbon molecule. Glycerol looks okay. Glycerol is this right here. So you've eaten a steak and it has a bunch of fatty acids, it has a bunch of triacylglycerols. Your body takes those triacylglycerols and breaks the fatty acids off and releases glycerol. Those 3 carbons of a glycerol molecule, those 3 carbons can be used for the production of glucose. Because remember, you have eaten a steak and you haven't put any barbecue sauce on it. All that you did was you peppered and salted it, and you're moving forward. You have ingested no sugar, but your body is going to compensate and figure out something to metabolize in order to make that glucose so that your blood sugar doesn't crash out. One of the sources is from those triacylglycerols. It will take that glycerol right there. The one that I was talking about up here. This one right here. And your body is going to convert that to this right here, dihydroxyacetone phosphate, through those steps that I showed on the previous page. Now, keeping in mind, you're not ingesting one triacylglycerol, you're ingesting hundreds upon thousands of them. So as soon as that glycerol is converted to dihydroxyacetone phosphate, what can happen to some of it? Well, some of it can be used to make glyceraldehyde. So just as you use glycerol to make DHAP, you can also use it to make GAP or GA3P or G3P. So this glycerol can be used to make this, which can then be used to make this. Once you have those glyceraldehyde 3-phosphate molecules and that dihydroxyacetone phosphate, you can make fructose 1,6-bisphosphate. Basically undoing the action of aldolase. Does that make sense? Okay. So once we've done that, then we just run our phosphatases and we regenerate our glucose. Now, with that in mind, my question a little bit ago was, well, which enzymes do we bypass? If we use glycerol, do we need to offset phosphofructokinase? Yes, we do, in order to get back to glucose. If we use glucose to make glycerol to make glucose, we also have to offset hexokinase. If we're using glycerol to make glucose, so going from here to here, or another way to ask this question is, if we're going from glycerol to glucose, are we ever going to make any 3-phosphoglycerate? This molecule right here. I changed the question, but it's sort of the same point. No, we're not. Because we're jumping in kind of midstream. Does that make sense? Was that an excessively roundabout question? It wasn't intended to be. Does it make sense now? Okay. So now kind of as a summary, we've talked about pyruvate to glucose, lactate to glucose, glycerol to glucose. We've got I feel like a doofus every time I go from like slide number 27 to 1 because of all these little animations. Like, ooh, what happens when I pop up this? And I have to undo like all 7,000 of them. So I feel like a doofus. Okay, so substrate number 1 was lactate. Lactate goes to pyruvate, pyruvate, done. Substrate number 2, glycerol. Glycerol goes to dihydroxyacetone. That plugs in to make glucose. Our third and last substrate is alanine. Now, when exercising, muscles produce large amounts of pyruvate. Some of this is converted to alanine by a reaction known as a transamination reaction with glutamate. Now, once alanine reaches the liver, the same transamination reaction reconverts alanine back into pyruvate. What I want you to know about this is that alanine, I would just say, utilizes a transaminase. Now, if I were to look at this and say, oh, okay, well, or if I were to take this, look at that. How about that, Joel? Good problem solving. There we go. If I were to take alanine and say, oh, where does alanine fit in on this? Alanine kind of doesn't fit into this. The only way that it does is if we drew it out here. Alanine goes to pyruvate through I'll label it as a TA or a transamination or a transaminase. Alanine doesn't go through some anything really fancy. It doesn't skip over anything. Pyruvate is made and converted to alanine. Alanine can flow through the bloodstream. Alanine can then be used to, when needed, go on to make glucose. So when we look at that glucose alanine cycle, or this process by which alanine can be used to make glucose, what I want you to know and what your important takeaway from this is that we're using a transamination reaction. I bring this up just because when you take metabolism, you will talk about transamination reactions because transaminases are central to amino acid metabolism. Now, the last thing is, well, there's energy consumed during all of this. 2 ATP consumed with pyruvate carboxylase, 2 GTP consumed with PepCK, and 2 ATP consumed when converting 3-phosphoglycerate into 1,3-bisphosphoglycerate. If you're looking at this and asking me the question, okay, that's all great, that's a lot of stuff. What's the important takeaway? I would start by looking at this. Being able to explain how do I go from pyruvate to glucose? Well, first I got to take pyruvate, put it in the mitochondria, and then rip it out of the mitochondria. And it's and how am I doing that? Well, I'm carboxylating it to make oxaloacetate. Oxaloacetate goes on to PEP. PEP goes through a series of reversible steps to get us F16BP. I use a phosphatase. I rearrange. I use another phosphatase. I'm at glucose. Um, I got a private message. Do I teach metabolism? Random question. No, I do not. It's a great question. It's a fantastic question. It's a fantastic class. You'll love it. Um, now, so I would start with this and just this is like the second time I've mentioned the movie Fight Club, I think. No, the second time I mentioned it today. I mentioned it in one of my other classes. Um, in the movie Fight Club, there's a scene or there's a series of scenes where they're reading from old Reader Digest magazines. Um, where it's like stories written by an organ from the organ's perspective. Like I am Jack's liver or something like that. I want you guys to do that, but say, I am pyruvate. What am I doing? Well, first, I'm going to go into the mitochondria. There's not a lot of hoopla with that. I can just go into the mitochondria because there's a protein that helps me move into there. Once I'm in there, then I'm going to get this nice little hat. Top of the morning to you. It's a carboxylate, or it's a carbon dioxide. Top of the morning to you. I've been carboxylated. Then I'm going to make my way out of the mitochondria. Again, top of the morning to you. I'll donate that CO2, but hey, nice trade-off. CO2 for a phosphate, I'll take it. Now I'm PEP. PEP gets back to fructose 1,6-bisphosphate, phosphatase, rearrange, phosphatase. So try to have that like perspective, or if you need to. Um, and then everything else beyond this is really like, because all of those other intermediates, if you, this is why, and this is my last statement that I'm gonna say. Um, the better you understand glycolysis, the better off you're going to be. Because if you can see how different things can either plug into or come off of glycolysis, that's going to position you really well. So then you can say, oh, okay, well, I mean, heck, if I can get it here, then all I have to do is this enzyme to get back to glucose. Okay, well, that's what I've got. If you guys have any questions, you're more than welcome to hang around. Um, this was a lot. Be patient with yourself. And remember, if there's like priority one, it's glycolysis. The better off you are with glycolysis, the better off you're going to be with everything else. Um, but other than that, thank you guys very much for coming. I hope you have a great day. No. I will get those squared. Does gluconeogenesis happen all the time, but in certain conditions have three pathways to fall back on? Um, Gluconeogenesis and glycolysis are kind of always taking place just because, and it really just depends on how much how much, what does the body need at any given point in time. I always kind of think about it as, yes, they are all, or they are always taking place, but sometimes the you're going to push the pedal down on one and release the pedal on the other. So they're always going on. It's never that glycolysis is not active at all. It's just when do you really need to lean in on one of them over another? Okay, I'm going to stop my share. Um, no, I had no poll questions today, so you guys are good. I apologize for that. Uh, Thank you very much. Okay, can I clarify the precursors really fast? Your precursors, lactate, glycerol, alanine. Lactate goes to pyruvate, alanine goes to pyruvate, glycerol goes to DHAP. I really didn't think that I could do it so fast, but I did. Okay, I'm gonna stop my recording.